Welcome back to the second part of chapter 12. Rest assured that this part is going to go a lot faster than the first part, and there's a lot less specific detailed information, and hopefully you'll enjoy it. So in the chapter, we've been talking about learning and memory, processes of learning information, consolidating it from short-term to long-term memory, the idea of short-term memory sort of changed to working memory and what that means, and of course, the changes that take place in terms of LTP and LTD, which are very minute and can be very annoying, but I tried to give you the overall take-home message and sort of the main ideas of this. The last part of this chapter, the last maybe 13 or 14 slides or so, is having to do with problems associated with age-related decline or genetic disorders that affect learning and memory. So we're going to talk about a couple of them here, and then I will give you a couple links with it to look at videos and stuff like that, like I did for the first part. You'll find it all under the chapter 12 lectures and links. So one of the things that happens, as we've talked about when we age, is that our DNA starts to break down. So problems with our visual acuity not being as sharp, hearing sort of goes down a little bit. And as our DNA breaks down, our bone density gets lower, our hair turns to gray, skin gets more wrinkled, and our, body starts to sort of break down. It's not as sharp as it used to be. We talked about in chapter 3 how the brain can repair itself the younger we are because of plasticity, right? The ability for our neurons to move and bend and shape and become stronger and weaker. So we can think of the ideas of LTP and LTD happening a lot easier when we're younger. But as we age, problems like strokes and age-related memory loss can lead to problems such as cognitive impairment and even dementia. There are different types of dementia. The one that is the most popular that people talk about is called Alzheimer's disease. So that's the one I will be talking about right now. So back in the day, researchers long believed that dementia in the elderly was inevitable. As we are breaking down, as we are aging, as our skin is wrinkling and our hair is graying and all of that, we are just bound to lose neurons. They're just going to die out. But what we see in a lot of people is that there are plenty of people and plenty of studies to show that although some neurons are lost, a lot of people show little to no memory loss, even as they approach over the age of 60 or even 70. Of the people that were tested, deficits in the ability to recall things or learn new things sometimes reflects motivation on memory tests. Maybe people don't have the motivation to try and remember. I always think of the average retired person, right? There is the type of person that you may see stereotypically down south that just wants to sit on their porch, watch the sun rise and set, watch life go by, and drink sweet tea on the porch or alcohol. You never know what's in the tea. But in that case, you're not really doing much to keep it trained, right? If you ever are a person who works out, one of the things that trainers and people will tell you is that if you don't use those muscles and you don't keep, engaging them, that you're going to lose your muscle mass and definition and stuff like that. Well, the brain is like a giant muscle. And a lot of times, those people who continue to use it afterwards, right? So retirement, they don't lose it as easily. Genetics may play a role in this, but for the average older person, right, a lot of stimulation, and I don't mean watching TV, although some shows may be more stimulating than others, but people that are retired that like to do Sudoku and crossword puzzles and continue to read about new things and discover new things, you're training your brain and you're working it out. And so although it may not be as sharp as it was when you were younger, that still helps to protect against some of this age-related memory loss. And so those people that have memory tests, sometimes if you're not motivated to keep using your brain, it may start to break down on you a little bit. Looking at studies of older rodents, this is what we see, that some of them may have some cell loss, but a lot of them are still able to do a lot of the things that they were able to do before. We don't really see a lot of loss of hippocampal neurons. Remember, earlier in this chapter, we talked about how the hippocampus is very important in spatial memory and in temporary storage and working with the frontal lobes and the cortex to help with working memories and things like that. We do see some loss of NMDA receptors, so LTP may suffer. Some of the cells may get demyelinated, not to the point of multiple sclerosis, but it may affect the efficiency of firing of neurons. And of course, in the prefrontal cortex, we may see a decline of cells as well. So this idea of using your brain so you don't lose it is what we call the reserve hypothesis. We know that exercise is good for the body. It's also good for the brain. So an active lifestyle throughout life, continuing to work out and continuing to use your brain is really good for it to continue firing. And it seems to be protecting against this age-related memory decline. Actually, in this reserve hypothesis seems to prevent cognitive declines, at least where it can. If you have genetic deficiencies for like a Parkinson's disease, which affects memory and, as we talked about in chapter 11, movement. If you have MS, as we talked about in chapter 3, that can also, cause neurons to die off because they're not getting the right amount of stimulation. And of course, genetic disorders like Alzheimer's disease are going to affect that. But all things considered equal, those who use their brain more, exercise it, and actual physical exercise tend to have a better prognosis later on. There in the chapter, it talks about these nuns and this idea XXX study. I'm going to provide a link to you for an article about that. But essentially, there were nuns that, were writing journals daily about their activities. They were asked to write journals about their day. And there were 4 nuns, 3 of whom actually developed Alzheimer's when they got older, and one nun that did not, and did not seem to have many declines. And when they went back through the paperwork of the journals that the nuns had written, one of the stories was that the 3 nuns that actually developed Alzheimer's, that had that cognitive decline, when writing about their day, they were very limited. They did not spend a lot of time on it. Oh, I did this, and then I prayed, and then I did this. And it was, basically just a few lines about their day. The one nun that did not get Alzheimer's disease, when they looked back through her journals, she wrote so many little details about everything and sort of elaborated on everything and really used her brain to talk about, instead of just saying, I talked to Father so-and-so, it would be like, I talked to Father so-and-so, we talked about this, this, and that. He really liked when I said this, and got really, really specific, really used her brain to write really specific details. And that seemed to at least show a correlation here of the not being susceptible to this dementia. But you can read more about that in the link. So looking at abnormalities of learning deficiencies and disorders, obviously we've talked about Parkinson's, we've talked about MS, but probably the biggest age-related memory decline disease or disorder is a type of dementia, a very specific type of dementia called Alzheimer's disease. And in Alzheimer's disease, what we see is that the brain begins to deteriorate. It gets smaller and smaller. It becomes a shrunken brain because the brain is made up of all of these different billions of neurons that start to die off. And when those neurons die off, the brain starts to shrink because that's the building blocks of the brain. So we see that there are deficits in language, visual spatial functions, the ability to reason, And a lot of the people with Alzheimer's disease, as it progresses, they get really disoriented and very aggressive because they're not able to do the things that they were able to, and they can't articulate that. It affects about 10% of the population of elderly people over 65 years of age. And as you get closer to 85, that number goes up to about 50% of individuals. That is a huge spike in the number of cases, but think about the sheer number of people that live to 85 as opposed to 65. So you're not getting a big increase in the overall number of people. You are getting a big increase in the percentage of people that live that long. And it makes sense. As the DNA in the brain is breaking down, there's more chances of this disorder in that population. What we see here on the bottom are two symptoms, physical symptoms that we see of Alzheimer's disease. So we're going to break this up into what are the clinical symptoms that you see. Obviously, the inability to remember things, problem recalling information that you once learned, not being able to recognize people that you know. Those are the clinical symptoms that we see. Those are things like that. But since this is a biopsych class, we're going to talk about what we see in the brain that leads to these clinical symptoms. So what we see here on the left in A, there are two different things happening. One with respect to the outside of the cell, and one with respect to what's going on inside of the cell. So there is a protein that our brain produces called the tau protein. And the tau protein may sometimes accumulate into bigger clumps. If we overproduce the amount of protein that we have of tau, then those tau molecules basically become what we call plaques. And just like plaque is no good for your teeth, it's sort of this black stuff that forms and tries to eat away at the enamel of your teeth, which causes cavities. We have plaques in the brain that are accumulated from too much of this tau protein, in overproduction of this tau protein. And so what it causes is it causes these plaques that attack the neurons. So from the outside, we have these plaques that may be attacking neurons and causing death of the neurons, cell death, from the outside. On the right, what we see is the inside of the cell. Now remember, the inside of the cell has its own processes. It's got its own mitochondria and all those little things inside. We have neurotransmitters that are being processed and synthesized, packaged in these vesicles, moved to the end, and right, so that has to be efficient as well. And there are a lot of processes inside the cell. Well, one of the things that happens is some of these lines get tangled. And if these lines are getting tangled, then things are not able to progress inside the cell. So on the outside, we have this tau protein that's making these plaques eating away at the neurons from the outside. And on the inside, it's like if you have these lines that get all tangled up. So they're called neurofibrillary tangles or neurofibrillary tangles. And what happens is the processes inside the cell get tangled up so nothing can get through. It's basically like you have a giant knot inside the cell, and so that causes cell death from within. So it's the combination of these plaques on the outside and the cells getting tangled up and dying off from the inside that cause all of the cell death, which leads to the aggressive and progressive nature of the disorder. By the end stage of Alzheimer's disease, people have trouble controlling their bodily functions and are really sort of in bad shape. It's not a very fun disorder at all. So you can see on the right is a normal brain, healthy brain, lots of nice healthy gyri, the bumps on the brain. And so what we see is because of these plaques and tangles and the problems associated with Alzheimer's disease is a global loss of neurons. We see this especially in the frontal lobes and the hippocampus, but you can see it deteriorating all over, as you can see from that picture on the left down below. Because we know the ventricular system is where the cerebrospinal fluid flows through. When you lose neurons around those, the ventricles become a lot bigger. So not only are we losing neurons, but if we have more um, or bigger ventricles, then you're going to have more cerebrospinal fluid that's kind of coming through and washing things away. Having the right amount of cerebrospinal fluid is really good for the brain, protecting it, throwing away the waste, bringing nutrients into the brain. But if you have too much going on and you have a lot less neurons, that can cause a really big imbalance as well. And the hippocampus, which had a lot of connections to the frontal lobe and to the cortex, is also isolated from the rest of the brain, hence learning new things, recalling things, right? If you're getting this big disconnect between the hippocampus and the frontal lobe and the prefrontal cortex, that's going to cause a lot of problems when you're trying to learn or recall information and things like that. So a lot of these memory problems are associated with cell loss, isolating the hippocampus, a lot of loss, as you can see here, in the frontal lobes. And it's those acetylcholine neurons that seem to be the ones that are getting destroyed, as we talked about in chapter 2. And those, like we said, besides the glutamate that we talked about earlier this chapter, acetylcholine inside the brain, not down at the muscles, is also directly involved with learning and memory, and those are the ones that are breaking down first and most. So we know that the clinical symptoms are the memory loss, the inability to function, aggression, and those things. We also know that physically in the brain, we see the plaques and tangles that are destroying neurons, plaques on the outside, tangles on the inside. And because this is a biopsych class, we also look for genetic correlates. Because Alzheimer's disease is a genetic disorder, your chances are more likely that if you had a parent or a family member, especially if it's immediate family, parent or grandparent, that suffered from Alzheimer's disease, your chances are increased compared to the general population. Does that mean you're going to get it? Not necessarily. And again, I think that when we talk about the reserve hypothesis and using the different techniques for trying to preserve the neurons that you have, it's a good idea. So if you have parents or grandparents that have recently been retired, and especially now in a place where we can't really leave the house very much, it's very easy to get complacent and to not do new things. One of the things that we see in the population that I'm part of, which is professors, especially when I was at UCLA, there were a lot of professors that went way past 65. The head of my department for behavioral neuroscience, his name is Frank Krasny, and my first year of grad school in 2007, 0607, was supposed to be his last year as the chair of the department, and he was going to retire and move to emeritus status, which just meant he would work, a couple times a week, maybe teach a class or two, and enjoy most of retirement while still being there on a limited basis. But man, you could not kick him out to save your life. By the time I was in my 6th and final year, I took a little longer because we had Dr. C and I wanted to finish the dissertation after he was born as opposed to before. But you still had Krasny in charge of everything. He was still the chair of the department, doing all of the extra things, running a lab, teaching classes, because he just wouldn't give it up. And we see this a lot. My neuroanatomy professor through the med school actually passed away at the age of 86 or 87 while he was still teaching. And what you find in that population of professors that are always doing research and always learning and looking at new things and the new studies is you see a lot less of this cognitive decline, a lot less of Alzheimer's, unless they have some sort of genetic correlate. So here we found, through genetic studies, we have found 4 genes that lead to the potential symptoms of plaques and tangles and that lead to Alzheimer's disease. 3 of them are more of an early onset, but again, we don't see that big a population before the age of 65. So these are the 4 genes, and the one on the bottom, the ApoE4, is the late onset one. So here you see one called the APP gene, which is in chromosome 21. which leads to Alzheimer's earlier. Could be anywhere from 45 to 66 years of age. But we see this in only about less than 0.1% of cases. But the APP gene is there consistently. We also have two versions of a gene called presenilin, presenilin 1 and presenilin 2. One is in chromosome 14, the other is in chromosome 1. Presenilin 1 shows a really early age of onset, 28 to 62, but again, a very small percentage of people. Presenilin 2, again, 40 to 85, also less than 0.1% of cases. The one gene that's really, really involved, and actually I worked in an Alzheimer's lab as a postdoc for a year, a little over a year, and ran tests with mice and with people, and we were looking at this ApoE4 gene. So the ApoE version 4, ApoE4 gene, is located in chromosome 19. It's associated with the later onset, so those above age 60, but we see this in about, in more than 50% of the cases that we find. And it's the ApoE4 allele that increase your risk anywhere from 3 to 8 times the normal population. And the ApoE4 seems to be directly involved with the ramping up of plaques and tangle formation in the neurons and in the brain. So can you find plaques and tangles without Alzheimer's? The answer is yes, just not in as big abundance. And so there are many ways that we can kind of look at this idea. Just like we talked about stem cells for possibly Parkinson's disease by replacing the dopamine neurons, there may be a chance that these acetylcholine neurons that are being destroyed in Alzheimer's can also benefit greatly from stem cell treatment by becoming more of the acetylcholine neurons within the brain. Come on. So what are some of the environmental correlates? Because again, it's not necessarily just genetic. Over 50% of the cases, but not 100%, right? So some of the things that have believed to have led to dementia, especially Alzheimer's disease, was lead exposure in children. There was a lot of lead paint that was used in facilities in the 40s, 50s, 60s. Pesticides like DDT that are even still used in some underdeveloped countries to try to protect the crops from food crops from bugs and things like that. The DDT definitely did a good job of killing the bugs, but the byproduct of the poison from the DDT may have caused these neural declines in a lot of people who may have consumed their products. Chronic stress seems to exacerbate a whole host of things, right? We always talk about trying to get stress under control. It's harder for some than others. But what about causes and treatments? We know that we don't have any treatments that can, other than potential stem cell research, that can replace the cells that were lost. But we also know, like in Parkinson's, we have too little dopamine. With these acetylcholine neurons that are being destroyed in Alzheimer's disease, we have too little acetylcholine. So in a similar way that L-DOPA is used to convert to dopamine and temporarily help people with Parkinson's, acetylcholine seems to help in the case of Alzheimer's disease. So one of the things we talked about early on in chapter 2 was talking about reuptake and reuptake inhibitors. For depression, we talked about a lack of serotonin, and so these newer generation antidepressants are SSRIs, and we talked about how SSRIs work by blocking reuptake or by blocking the enzyme that that breaks down the neurotransmitter. Either way, leaving it in longer. So obviously the SSRIs were the ones that blocked reuptake, and then we had ones that blocked the monoamine oxidase, which is what broke down the monoamines. So we call those MAO inhibitors. In the same way, very much the same way that MAO inhibitors work, the treatment for Alzheimer's disease is to try to inhibit the enzyme that breaks down acetylcholine. So with acetylcholine, we use what are called, that enzyme, by the way, is called acetylcholine esterase, or ACE or ACHE. So we call them ACE inhibitors, ACE being acetylcholine esterase. That's the enzyme that breaks down acetylcholine. Sometimes it's actually, even though glutamate is good for learning and memory, too much glutamate, though, may be a bad thing, because too much glutamate may saturate your receptors and kill the NMDA receptors by overstimulating them. So another drug called memantine, or the generic name is Namenda, actually helps to reduce levels of glutamate to try to preserve those NMDA receptors and not kill them off. Of course, they've tried drugs that try to do things like break up the tau protein and remove the plaques, or try to remove the tangles. And they've had some success, but mostly they're ineffective. So attacking the plaques and tangles doesn't necessarily work, although it's a great idea when looking at it. Actually, there is a great deal of information that says a lot of inflammation in the brain, just like inflammation in the body can be bad for people. Inflammation in the brain could also be leading to Alzheimer's. So there was when I was in this lab about 7 years ago, there was a promising treatment by using actually a similar byproduct of the why am I drawing a blank on the name of the little blue pill that helps with erectile dysfunction. I don't know why I'm drawing a blank right now. You guys know what I'm talking about. And so in that way that it causes more blood to flow out of the brain and into the penis, that can also, as a byproduct, reduce inflammation because you're reducing blood flow and things like that. That one, I don't know if it ever worked out because I never read anything in the journals, but that was sort of the new treatment that they were about to start looking at when I left the lab. So just to let you know. It used to be that you could not confirm, before we had updated brain scans and things like that, even as far back as 20 years ago, that you couldn't detect Alzheimer's disease until after death. You could say it was dementia, a progressive dementia. You could suspect that it was Alzheimer's disease, but it could never actually be determined until after death when you looked at the progression of the disease by looking at the brain basically after death. Now, brain scans, PET scans, and MRIs can reveal areas that are losing neurons more rapidly than others. Temporal lobe areas and hippocampus around there, parietal areas, frontal lobe areas. So we have gotten a lot better in the last 20 years in being able to look at the specific progression of cell loss and the pattern of it. And we could say with much more certainty that it seems to be the progression of Alzheimer's disease as opposed to another type of dementia or another deteriorating brain issue like Parkinson's. We also can look at biomarkers for plaques. Again, this tau protein that seems to be ramped up and causes the plaques that attack the neurons. This is also the same thing we see with multiple hits to the head. So if you think about football players, Junior Seau, if you know who that individual was, played for many years in the NFL as a linebacker, a lot of hits to the head and body, of course. But a lot of times we have seen in scans and things like that multiple hits to the head can also cause tau protein to overproduce and cause some of these plaques. So that disorder that's related to football players and repeated hits to the head is called CTE, chronic traumatic encephalopathy, which just means lots of hits to the head. But we see that plaques and tangles, or at least the plaques can build up, and you can see similar symptoms in those people. But looking at the biomarkers for plaques seem to be pretty promising. If we can tell early on that tau is going to or starting to overproduce, which may cause plaques in the brain, that early intervention is a better idea to help to prevent it, or to at least maybe stop it earlier. Because what we see is about a quarter of individuals, 25% of individuals that have plaques early on seem to contract Alzheimer's within about a 3-year period. And actually, there are people that have lower levels, too low levels of tau protein that may correlate to Alzheimer's in about 2% of patients. But 2% is not necessarily a lot to go on. It's definitely more evidence for too much tau than having too little tau. And of course, like I said, if we compare this to using your brain, this idea of the reserve hypothesis is that high idea density, having a lot of ideas, reading a lot of new books, doing Sudoku and crossword puzzles, even watching shows like Jeopardy and getting your trivia game on, shows that it may delay the onset of symptoms. So again, your brain is like a muscle, and if you use it, you're causing more LTP, you're preserving those connections in the brain, maybe causing more connections, and it's really doing a good job to protect against some of the problems that we see in Alzheimer's. Is it going to work for everybody? No. If you have that allele of the ApoE4 gene, right, that's going to be something that's going to be hard to overcome. But there are things that people can do to try to prevent cognitive impairment, or even, you know, impairment that may lead to what we see in Alzheimer's disease. Like I said, with the Roman Catholic study, 80%, so maybe that's 4 of the 5, low-scoring Roman Catholic nuns, when looking at their journal entries, eventually contracted Alzheimer's disease. And the one nun that had a really verbose, really wordy, specific entries in her journal, using a lot more of her brain and coming up with words and phrases, seemed to have protected against it. So lifelong learning, which we professors sort of dedicate our lives to, increases our cognitive capacity and may even increase neurogenesis, the formation of new neurons, which we don't see very often. If you recall from chapter 3, hippocampus and olfactory bulbs are the main places where you see them. But it seems to potentially provide a protective effect against dementia. So I'm going to take that to the bank with me and hope that I continue to be able to protect myself in later years. I'm not that old yet, people. Another progressive disorder of the brain that leads to memory loss and basically symptoms that are very similar to dementia is called Korsakoff syndrome. But Korsakoff syndrome is actually a progressive disorder that comes from too much alcohol. And a lot of people think that it's that the alcohol just causes the brain to shut down. But what it really is happening is that a lot of people are getting their calories from the alcohol and not actually eating. So they're getting a deficiency in certain vitamins and nutrients that they need. Specifically, a lot of foods are rich in vitamin B and B1 at that. So when people are drinking, not only are they obviously destroying neurons and hurting their liver, but they're also denying themselves vitamin B, and they're becoming vitamin B deficient. And what that does is it causes damage to parts of the brain in the memory system, not the hippocampus specifically, but something that the hippocampus is connected to called the mammillary bodies, which seem to contribute to memory formation. And if the mammillary bodies and this medial part of the thalamus are damaged, their connections to the frontal lobes are lost. And so you see a very similar progression of memory decline, similar to Alzheimer's disease, due to specifically drinking, which is, again, a progressive, irreversible deterioration, almost always caused by chronic alcoholism. And so here we see the alcoholic consumes large quantities of calories in the form of alcohol, and so they don't eat instead, they just drink. And so they don't get the adequate nutrients and vitamins in a diet. And it reduces, the alcohol itself reduces the absorption of thiamine, which is that vitamin B1 in the stomach, which causes the breaking down of these areas of the brain, the mammillary bodies, the thalamus, and it causes these effects. But It is not the same as Alzheimer's disease in cause, drinking, excessive drinking, and some of the cognitive effects are similar, but there are some differences. We see anterograde amnesia in declarative memory. So we see memory loss very much like Alzheimer's disease. And kind of like HM, we see an intact non-declarative memory. So it doesn't seem to affect, right, it doesn't seem to affect the basal ganglia, the striatum. It doesn't seem to affect the cerebellum, although, of course, drinking shuts down the cerebellum, so your balance is off, but not completely. It doesn't deteriorate it. But one of the biggest differences in Alzheimer's and Korsakoff's is this idea in Korsakoff's that it causes what we call confabulation. And confabulation is sort of making things up or thinking that dreams or things they saw on TV were incorporated in real memories, because without these mammillary bodies and the progression of this disorder, people get a lot more confused between reality and memories. So we see this inability to distinguish reality from fiction. You don't see that as much in Alzheimer's disease. So that's it. I am going to also include a video that talks about the similarities and differences between Alzheimer's and Korsakoff's. And I hope you enjoyed chapter 12. And what this means is, after this, we have one more week. You will have a quiz next week on this chapter, chapter 12. And then we just have the final paper and the final exam during finals week. I know it's been a long, arduous journey, but thank you for sticking it out with me, and we will get through this together. And as always, I will talk to you soon.